is Kristee Brown, and she will be presenting on studying the role of, Jak Stat3 Pathway in macrophage polarization and pancreatic tumor progression.
Good afternoon, everyone. My name is Kristee Brown, and this summer I was able to participate in the Community College Research Fellowship Program, in which I worked in the lab of Marina Pasca-Di-Magliano alongside mentor Ashley Velez. And this summer we took a look at the role of the Jak Stat3 pathway in macrophage polarization and pancreatic tumor progression. So pancreatic cancer is a very deadly disease. Currently, it's the third leading cause to cancer-related deaths, with a 9% 5-year survival rate. This is due to poor prognosis and non-effective treatment therapies at later stages. So it's very important for us to study the biology behind the disease to find better therapeutic treatment. So just some information, we find that KRAS mutations are found in over 90% of patient samples. We also see that KRAS drives formation and pancreatic cancer precursor lesions, along with requirement for progression and maintenance of the disease. And in mice models, we see that mutation of the p53 suppressor gene also accelerates formation. Here we have a visual of the effects of the KRAS mutation. So here we have our normal acinar cells, and these acinar cells function to secrete digestive enzymes through these ductal cells. And here we have an H&E staining, and here all in pink we have our abundant acinar cells. And here we also have our ductal cell. But upon initiation of KRAS, we see that these acinar cells start to develop these precursor lesions. These precursor lesions form these ductal-like structures. And you can see those ductal-like structures here in our H&E staining. We also see some early penetration of the immune system. But upon KRAS and p53 mutations, we find that we have a full formation of our tumor cells. And here throughout the pancreas, you see those tumor cells all throughout the pancreas. And we also have some stroma here. The stroma is actually filled with some immunosuppressant immune cells, such as the CD8 T cells and macrophages. So here we have a multiplexing staining that shows the microscopic view of the microenvironment within pancreatic cancer. So here we have our tumor cells outlined here in white. We also have some T cells here stained in green, and then we also have arginase 1 that are stained purple, along with an abundant amount of macrophages that are stained in orange. And we also see an insufficient amount of CD8 T cells throughout this environment. And you see one here, and we also see one here, but there's very few. But why is this? There's some tumor and immune cell interaction happening. Our CD8 T cells normally kill cancer cells, but our tumor cells have the ability to secrete cytokines. These cytokines then alter our macrophages, then allowing them to now secrete immunosuppressant proteins such as arginase 1 and YM1. This then inhibits those CD8 T cells from penetrating to the tumor cells, allowing for progression of the tumor cells within pancreatic cancer. So again, I mentioned that we have some change in the macrophages. So our normal anti-tumor macrophage functions or secretes the protein ionized. But after polarization and change, we start to see that these macrophages are secreting now arginase 1 and YM1, which are both immunosuppressant proteins. These immunosuppressive proteins now make these macrophages pro-tumor, inhibiting again those CD8 T cells from infiltrating the tumor cells. So in whole, we see that During this stroma of this microenvironment, of this immunosuppressant microenvironment, we have the JAK-STAT3 pathway that's highly activated within that stroma. Here we then ask ourselves, does the JAK-STAT3 signaling regulate myeloid cells' function during pancreatic carcinogenesis? So just some information about the JAK-STAT3. We see that STAT3 signaling promotes initiation and progression of human cancers, and this is by inhibiting cell death and inducing cell proliferation and invasion. We also see that the STAT3 full knockout mice have shown to protect against those early precursor lesions. And JAK-STAT3 pathway is highly activated through cytokines, as mentioned earlier, specifically IL-6, and this is known to be highly active in those precursor lesions as well as through pancreatic cancer. So the JAK-STAT3 pathway works when a cytokine attaches to the JAK cell membrane. This then allows for phosphorylation of the JAK protein. Phosphorylation of the JAK protein allows attachment of the STAT protein. And then after phosphorylation of the STAT protein, we see that a dimer is produced that then penetrates the nucleus, allowing for transcription to happen. So here we have in our experimental model in which we used a STAT3 flux lysine breeding. And this breeding allowed for us to then use a Cre-LoxP system to deplete STAT3 genes from myeloid cells. We then injected pancreatic tumor cells within this model. Within 2 weeks, we then dissected the pancreas, and what we found was that myeloid cells did decrease the tumor sizes of these pancreatic tumors within the mice. Our controls show here, compared to our heterozygous, along with our depletion of the STAT3 gene, was a decrease in tumor weight. We also have a CyTOF, which actually uses antibodies to detect cells along the cell membrane. And what we see was that there was some change compared to the control, heterozygous, and the STAT3 depletion mice, a change in infiltration of particular cells, or cell proteins, sorry. So in conclusion, we see that the STAT3 depletion did allow for a decrease in the weights of the tumors within the pancreas. We also want to do future studies to show the infiltration of T cells, macrophages, and other proteins involved in immunosuppressant environment within pancreatic cancer. So I just want to give an acknowledgement and thanks to Marina Pasca Di Magliano and lab, also my mentor Ashley Velez, UROP, CCSFP, and then also the Rajo Cancer Center. Thank you, and I'll now leave the floor open to any questions.